Now we are discussing about nucleic acid. In that we are discussing about the nucleotide structure. As we know that nucleotides are made up of three parts: phosphate group or a phosphoric acid or a nitrogenous base and a sugar. It can be the ribose or the oxyribose. As here you can see that it consists of the phosphate group and the sugar. This sugar is the pentose sugar. Okay, so it can be the ribose or the oxyribose. And when talking about base, it can be the purines or pyrimidines. Purines are adenine and guanine, and pyrimidines cytosine, thymine, and uracil. If DNA is the building block of life, then the nucleotides, these are the building blocks of DNA. Okay, so but what is a nucleotide exactly? So this nucleotides are a class of organic compounds that make up nucleic acid, the substance that defines hereditary traits of all living organisms. Nucleotides are an essential part of DNA, RNA, and cell function, and they can serve many purposes depending on their structure and chemical compounds. Okay, so here we'll go over the nucleotide definition and the different types of nucleotides out there, what makes each type of nucleotide unique, and why nucleotides are involved in nearly all cellular activities. Okay, so first, before we give you the nucleotide definition, here some helpful definition of words we will discuss. First, we will see of DNA, what actually it is. It's a deoxyribonucleic acid, a self-replication material present in nearly all the living organisms. And this DNA can be found in every cell of your body and is what carries all your genetic information. You've probably heard that it takes the shape of a double helix. Now coming to the RNA. RNA, ribonucleic acid, a nucleic acid present in all living cells. Its primary function is to carry instruction from DNA to synthesize protein, in other words. RNA is a middleman, you can say. Genetic information flows from DNA through RNA to proteins. Then purines and pyrimidines, a basic crystalline compound that forms hydrogen bar. They're the two basic type of nitrogenous base that form nucleotide bases found in DNA and RNA. Now coming to the monomer. Okay, so it's a molecule that can be bounded to other identical molecule to make a polymer. So remember this, that the prefix mono means one. And coming to the polymer, polymer is nothing but a substance made primarily or entirely of similar units bonded together, and each of this molecular unit is a monomer. And here the prefix poly means many. Now then, what is a nucleotide? Let's take a look over it. So nucleotide are the organic molecules that serve as a basic structural units for DNA and RNA, or you can say basic structure of monomers. Okay, structure units. So which, as we know, are the building blocks responsible for all life on Earth. And this each nucleotide contains a nitrogenous base and a 5 carbon sugar and at least one phosphate group. When bonded together, nucleotides create nucleic acid. That is the string of DNA. So the nucleotides can also stand independently and interact with cells in other ways. Okay, so let's discuss about the structure of nucleotide. So we know that nucleotides are building blocks of DNA and RNA, but they also do a lot of other things. In order to understand why and how nucleotides perform their extremely important jobs, let's first go over what they are made out of and how they become the nucleic acid. Okay, so first we will see of the nucleotide bases. So the type of nucleotide is defined by its chemical base. So generally there are 5 chemical bases, that is adenine, cytosine, guanine, thymine and uracil. The base and the amount of phosphate residue define how the compound is named. So with the help of an example, we will see an adenine nucleotide with one phosphate group is called as adenosine monophosphate. So here this adenosine, it will refer to the adenine or the chemical base of the nucleotide, and monophosphate, it will refer to the fact that it has one phosphate group, and remember that mono means one. These bases are each defined by a letter and are either pyrimids or purines. So first we will see of adenine. It will denote with the letter A. Adenine is a purine with a chemical compound C5H5N5. An adenine base nucleotide is called adenosine. Adenine is formed by 2 hydrogen bonds which help stabilize nucleic acid structure. ATP, adenosine triphosphate, is also an important form of energy found in most cellular function. Cytosine, it is denoted by C. Cytosine is a pyrimidine with a chemical compound C4H5N3O. Cytosine based nucleotide is called cytosine. Cytosine is a heterocyclic aromatic ring with 2 substituents attached. Cytosine, it pairs with guanine to form nucleic acid, but as a free nucleotide. Can work as a coenzyme that helps convert ADP to ATP. Coming to the guanine, it's denoted by G. Guanine is a purine with a chemical compound C5H5N5O. A guanine-based nucleotide is called a guanosine. Guanine is a fused ring with conjugated double bonds. Guanine bonds with cytosine via 3 hydrogen bonds to form the nucleic acid in DNA. Whereas thymine, it's denoted by the T. Thymine is a pyrimidine with the chemical compound C5H6N2O2. A thymine-based nucleotide is called thymidine. Thymine is a fused ring with conjugated bonds. Thymine bonds with adenine to form nucleic acid. This will help stabilize the nucleic acid structure. And about uracil, it's denoted by U. Uracil is a weak acid with a chemical compound C4H4N2O2. A uracil-based nucleotide is called uridine. Uracil is a dimethylated form of thymine and replaces thymine in RNA. Demethylation is a chemical process in the removal of methyl group from a molecule. The bases can combine with phosphates and sugar depending on how they are formed and serve as free nucleotide, in which they affect cell function, or this nucleotide can bond with one another based on their molecular structure to form the nucleic acid. Coming to the pentose monosaccharides, that's nothing but a simple sugar. Each nucleotide is a molecule, so while the bases are extremely important for how the nucleotide is classified and for its eventual function, they cannot form without the other element that make up the molecule. So one of this element is simple, that is 5-carbohydrate sugar. A nucleotide can contain one of two sugars, deoxyribose, a monomer of DNA, or ribose, it's a monomer of RNA. Just because the nucleotide base has one of the two type of sugar, that doesn't mean that it will necessarily bond with other nucleotides to form the nucleic acid. Okay, so here coming to the phosphate group. Phosphates are the chemical derivative of phosphoric acid. You might have heard about the phosphate when discussing certain household items, right? Inorganic phosphates are used in things such as fertilizers and laundry detergent. Naturally occurring phosphates, however, are an integral part of the formation of nucleotide. Each nucleotide is made up of 1, 2 or 3 phosphate group. Free nucleotide can be made up of a sugar, a base and 1 or 2 phosphate group. Therefore, they are known as either monophosphate if it has one phosphate group, or it can be the diphosphate if it has 2 phosphate group. So the nucleotides that bond together to make the nucleic acid in DNA and RNA are triphosphates. It means they have 3 phosphate group. Nucleotides are coded by the sugar, the base and the number of phosphate group. For example, a nucleotide called ATP, right? A nucleotide like DATP is there, that is nothing but deoxyadenosine triphosphate, while GMP is guanosine monophosphate. So if the name does not have D in it, then this indicates that it is made from a ribose sugar instead of deoxyribose sugar. So here, what do nucleotides do? So we have already gone over the nucleotide definition, but what exactly do nucleotides do? In other words, what is their purpose? DNA and RNA. We know that the RNA and DNA are made up of strings of nucleic acid and carry out genetic coding. That is, RNA and DNA are changing all the times, and the cells are constantly growing and dying in them, as well as in all the other parts of our body. So nucleotides are a major part of this process in a few key ways. First, they form that basis for nucleic acid. Second, working outside of nucleic acid, they help trigger and even participate in cell function. In order to perform nucleic acid, 2 triphosphate nucleotide must bond via hydrogen atoms in a process known as base pairing. So each base is formed by complementary nucleotides, one purine and one pyrimidine. Purines adenine, guanine, whereas pyrimidines cytosine, thymine and uracil. In terms of nucleic bases, here are the triphosphate that make up DNA. This is DATP. It's nothing but deoxyadenosine triphosphate, a nucleotide that is made up of deoxyribose sugar and adenine base and 3 phosphate group. DCTP, deoxycytidine triphosphate. It's a nucleotide that is made up of deoxyribose sugar, that is cytosine base, and 3-phosphate group. Whereas DTP, it's DTTP, is deoxythymidine triphosphate. It's a nucleotide that is made up of deoxyribose sugar and a thymine base and 3-phosphate group. So the nucleotide that make up RNA are ATP, adenosine triphosphate, a nucleotide that is made up of ribose sugar and our adenine base and 3-phosphate group. CTP, cytidine triphosphate, a nucleotide that is made up of ribose sugar, a cytosine base and 3-phosphate group. Whereas GTP, guanosine triphosphate, a nucleotide that is made up of ribose sugar, a guanine base and 3-phosphate group. UTP, uridine triphosphate. It's a nucleotide that is made up of ribose sugar, uracil base and 3-phosphate group. So for example, DCTP and DGTP bonded together would form a nucleic acid. Coming to the free nucleotides, di and monophosphate nucleotides cannot bond to become nucleic acid. Nevertheless, this nucleotide still have important cellular functions. So the nucleotides can act as coenzymes. An enzyme is a substance that is produced by living organism and that act as a catalyst to bring about a specific biochemical reaction. So they can help speed along chemical processes when bound with an enzyme. So the function of the coenzyme depends on several factors, including what the nucleotide bonds with. So ATP is particularly served as coenzyme frequently and is considered the main energy currency in living cells. So since ATP is so stable, it stays in a cell until it's ready to be used, and then it releases energy to trigger a chemical reaction. So the nucleotides also play an important part in cellular metabolism. So this is a process that takes place in cells in which the cells are degraded due to the chemical reaction in the nucleotide. So here this process is especially important in RNA and DNA as it's happening within our cell at all the times. It means that it's extremely important. It goes right. If not, it can result in a variety of diseases. So this reaction is triggered and the nucleotide and the cellular degradation begins. So when this occurs in RNA and DNA, sometimes parts of the nucleotide can be salvaged to create a new nucleotide. So overall we can say that nucleotides are just one part of the intricate world of cellular biology. They play a central role in the life and structure of DNA and RNA, and their function is incredibly important in the build-up and breakdown of cells. So we can say that our cells are carefully working together every day, and understanding what a nucleotide does can help us make sense of basics of our cells and how they work.